Hi, my name is Saneem, and here's my SciFair project. It's on the use of concentrated oxygen as a microbial inhibitor for antibiotic-resistant bacterial infection, and this is a second-year study. My first-year study was based on if the concentrated oxygen would work as a bacterial inhibitor, and it was found that controlled bacteria were able to be inhibited by this concentrated oxygen. So this year, I wanted to see if it would be applicable to real-life studies by testing it on real-life environmental bacteria. My question was, can we inhibit the growth of Escherichia coli, methicillin-resistant Staphylococcus aureus, and other unknown antibiotic-resistant bacteria by increasing oxygen concentration to use as the microbial inhibitor? And my second question is that will it be significant enough to be applicable to real life situations? The reason I came across this question was because there's a real problem in third world countries about nosocomial infections. These nosocomial infections are bacteria that are getting into patients' bodies in the hospital. Basically, they're bacteria that you acquire in a hospital. And this is a great big problem in third world countries. So I wanted to do this project to find some hope for that. My hypothesis was that the increasing concentrations of oxygen will inhibit the growth of the various antibiotic-resistant bacteria, therefore acting as a microbial inhibitor. My second hypothesis to my second question respectively was that the data will be significant enough to apply to real-world situations. And my null hypothesis was that the different concentrations of oxygen will have no effect on the growth of the antibiotic-resistant bacteria. The materials I used were 31 McConkey plates, the 31 chrome agar plates, and 31 sheep blood agar plates. The rest here aren't very significant because they're just the bacteria that I got from the environment. Then I used swabs, which I used to pick up the bacteria from the environment. And then here are the concentrations of nitrox that I use. Nitrox is a mix of oxygen and nitrogen, which was tested in my procedure. The independent variables in this project were the amount of oxygen percents used in the nitrox. The dependent variables were the amount of bacterial growth that were on the plates. And the control group was the 35% oxygen that showed a lot of growth. And the constants were the areas that were swabbed, and these were constant between the three different types of plates. The procedure was repeated 10 different times for the 10 different areas, and all with the different plates. Here's the procedure for my project. I was lucky enough to work at the local hospital's lab, and there we conducted this experiment. Here's as follows. In the lab, we went and prepared swabs, and we prepared enough to go to 10 different places with 3 different swabs each for the respective place. When we went to the unique place to swab inside the hospital to get the environmental bacteria, we would swab the area for a good amount of time, and then we would put the swab back into a very sealed container and bring it back to the lab. When we came back to the lab, we had 30 swabs, 10 from 10 different places, 3 from each place. When we came back, we had plates for us. And these plates were the McConkey plates, the chrome agar plates, and the sheep blood plates. And we would put 3 from each respectively. And once we would have the plates, we would prepare them and swab them in the quadrant method, meaning we would swab half the plate, then we would turn it 90 degrees, and then swab, and then turn until all 360 degrees were met. Once we were done swabbing all the plates, we would incubate them for 24 hours. Once we incubated them and then came back to check them, we looked at the results to see how much bacteria has grown. And we got very different results. But for the few that were being tested with, we sprayed with the varying concentrations of either 35% nitrox, 50% nitrox, or 65% nitrox. And these different kinds would give us the data that we have here. Okay, so here was the data from last year's experiment. This showed that the types of oxygen were effective in fighting the uptake of bacteria. And the red means that there was no uptake, and the green means that there was. Over here, you can see that as the plates were moving to 65% efficiency, 65% nitrox, the amount of bacteria that were not present increased. And the green means that there was a lot of decrease. The yellow means that there's some decrease, and 0 means that it's constant. Either there was no growth throughout, or there was the same growth throughout. Here from the charts, you can see that there was 23 out of the 30 that grew bacteria in oxygen, the 35% oxygen, which is the regular oxygen that we breathe. This was our constant, or our control. The 50% oxygen, there was 12 out of the 30 that grew, and when we reached the 65% oxygen, there were only 4 out of the 30 that grew bacteria. And this goes to show that the oxygen actually prevented the bacterial growth, and it can be applicable to real-world situations.